I'm Marcus Kronforst. I'm a professor in the Department of Ecology and Evolution. Our research in general is really focused on the genetics and evolution of sort of these general evolutionary processes. So thinking about like adaptation, how organisms evolve and adapt to their environment. We also think about where new species come from, so the process of speciation.
I'm Nick Van Kuren. I'm a research scientist in the Kronforst lab in the Department of Ecology and Evolution. Our lab is really focused on like the evolution and development of butterfly wing color patterns and behaviors. And some really unique systems in these butterflies involve situations where males and females can look completely different. They can have completely different wing color patterns.
The species we're focused on is this perfectly delicious butterfly, Papilio alphanor, and it has evolved to look like other distantly related butterflies that are toxic to gain protection from predators. And supergenes are sort of a historical phenomenon, and this refers to when you get these complex phenotypes that are polymorphic in a population, but they're controlled by just a single locus, just one spot in the genome that changes a big, broad suite of traits.
So what this study was focused on was identifying not only the differences between the two versions of the gene, the alleles of the gene, but also how those affected how the gene functioned and turned on this novel mimetic color pattern or not. So this study was focused on using new genomics, functional genomics tools, and methods like CRISPR-Cas9 to really dissect the molecular functions of this mimicry supergene.
Years ago, what got us interested in this particular system, trying to figure out, okay, what molecularly is responsible for a supergene? And so over the years, we dug in, found out that unlike most other supergenes, where it's like a large part of a chromosome that's been captured in one of these supergenes, tens or hundreds of genes, in our butterfly, it's in fact just this one gene, double sex, that has made the supergene. It is inverted, it's flipped, so it's got sort of this classic supergene phenomenon. And now Nick's work and other people in the lab have gone in and actually dissected using sort of these modern tools of functional genomics and using CRISPR, further dissected that inversion to figure out what parts of it are actually functional. So that's why I think this work is very, very important, because we're getting to the point of dissecting these things that historically have been kind of intractable.